We need to have a really great understanding about cells in order to understand how the body works. So that's what we're going to talk about here. The body is made up of trillions of cells, and the cells come in different shapes and sizes. For example, some cells are nerve cells that have to go all the way from basically your tailbone to your great toe. So that cell is about 3 feet long. So it's kind of hard to imagine that, isn't it? But it makes us appreciate that cells are going to be in all different types of shapes. And the function of the cell is going to be determined by sort of the shape or the anatomy of the cell. Here it comes. We're talking a bit more about chemistry, but cells are composed of 4 elements mainly, and here they are listed, and they're composed of about 60% water. In fact, our body is made up of about 60% water, which kind of makes sense. And that the cell has 3 main parts. You may remember the nucleus is the innermost part of the cell. The cytoplasm is a jelly-like structure outside of the nucleus, and then we have an envelope that contains the entire cell, known as the plasma membrane. Let's talk about the nucleus. We do think of it as the control center of the brain because it contains deoxyribonucleic acid, which is also known as DNA, nice and short. The DNA is like the blueprint for the cell on how to build proteins, for example, and how to reproduce the cell. So it is sort of like the recipe card for a recipe. It tells how things are going to be built. So that's why DNA is very important. Also, when we look at the nucleus, we see that it has its own little membrane or nuclear envelope. So it has its own membrane. It has a nucleolus inside of it, which we're going to be learning about, and it has chromatin. The nucleolus has a double membrane called the nuclear envelope. It has little openings called pores so that things can move in and out of this area. Also, it has its own jelly-like fluid called nucleoplasm. So that's different than cytoplasm, isn't it? Because in this case, it's inside the nucleus. Also inside the nucleus, we have a nucleolus, and this is where ribosomes are made. They can move out of this area through the nuclear pores into the cytoplasm. Inside the nucleus, we also have chromatin. Chromatin is made up of DNA and is scattered throughout the nucleus, and it's present when the cell is not dividing. However, things change when it's time for the cell to divide. When the cell is about to divide, this DNA condenses to form these rod sort of structures called chromosomes. We need to take some time to really learn about the cell membrane called the plasma membrane. It is made of a phospholipid bilayer. So let's take a look at that now. It is considered transparent. It separates the contents of the cell from the outside of the cell. It has 2 layers of phospholipids arranged tail to tail. Okay, so we're going to look at that in the next image and how that structure works. There are cholesterol and proteins scattered along the phospholipid bilayer. And then there are sugar groups attached to some of the phospholipids, forming a thing called glycolipids. Okay, this may seem a bit overwhelming. Don't worry, we're going to look at it in the next slide. Structure is amazing to me. So again, this is the cell membrane or plasma membrane. It forms the outside wall of a cell, and it keeps the contents of the cell inside for the most part, and keeps the outer world outside from the cell for the most part. What we have is a phospholipid bilayer. We see all of these round little heads with tails going inwards into the cell membrane. So there are 2 layers of that. There are all of these heads on the outside with tails pointing inwards, and on the inside of the cell membrane, again, we have all of these heads with tails pointing inwards. That's why they call it a phospholipid bilayer. Then embedded in this phospholipid bilayer, we have some proteins, and some of these proteins extend throughout the cell membrane. It's important to understand that because we're going to build on that knowledge. These proteins can act like little doors that can open and close and allow things to move in and out of a cell. So it's really important to understand sort of the anatomy of the cell membrane, so then we understand how it works. Also, I want to mention that the tails are called non-polar, and they line up on the inside layer of the phospholipid bilayer. And the heads are called polar, and they are on the outsides of the cell membrane. Also, let's point out that we have some sugar groups attached to some proteins, and we call that glycolipids. They look green in this image, so glycolipids can be stuck onto a protein or onto the heads of the phospholipid bilayer. Talk about this layer some more. We really need to understand it well. So we have hydrophilic, that means water-loving, polar heads. They are on the outside surfaces of this membrane, and outer surfaces, I should say, of the membrane, because there's an outer portion, and then facing the inside of the cell, we also have these polar heads. And then we have hydrophobic or water-fearing, non-polar tails form the center of the membrane. This interior makes the plasma membrane relatively impermeable to most water-soluble molecules. This is important because some of these molecules do need to cross this membrane somehow, but we can see now that it's pretty hard for them to cross. So they need special ways of crossing the plasma membrane, which we're going to learn about. The role of the proteins in the plasma membrane is very important. They are able to recognize when a hormone comes along, for example, or another chemical messenger comes along at the surface of the cell membrane. Some of these proteins can recognize specific chemicals and hormones. Also, these proteins can act like channels or carriers to carry things across the cell membrane that need to be carried. They can be opened and closed like doorways, for example. Sometimes I'll joke around in class about how the cell membrane looks like a butter sandwich with olives piercing through it, if that helps at all. A butter sandwich has two pieces of bread with butter on the inside, and we could have olives studding this sandwich, going all the way through both layers of the bread. An olive has an opening in the middle sometimes that could allow the passage of some things. So if that helps you at all, then that's great. Otherwise, you may just think it's silly. Take a moment to talk about how cells butt up against one another. There can be tight junctions between cells which create a waterproofness, so a leak-proof sheet. Also, cells can have desmosomes between them, which are like welds or rivets between two cells to hold them against one another. It calls them anchoring junctions here. They're really important in the heart, for example, because the heart muscle cells contract, and we want them to really securely anchor to one another so that they don't come apart from each other. Between cells, there can be gap junctions. These are like little tubes that span the distance between two cells, and it allows molecules to move from one cell to the next. This is used sometimes for sending a signal between cells more quickly. This slide is showing a lot of what we've learned already. In the middle, we have a cell, and the cell has yellow fluid called cytosol. It also has a nucleus. I believe they're trying to show that in purple here, the large purple structure in the middle I would call the nucleus. On the top of the cell, we have microvilli. Microvilli increase surface area. Okay, also, how do cells stay enclosed? Well, they have a cell membrane, so the whole outer edge of this cell is enclosed in an envelope called the cell membrane. We've learned that it's also called the plasma membrane, and it's made up of a phospholipid bilayer. Okay, so how do cells connect to each other? What happens there? Sometimes there are tight junctions, which can make a sheet of cells waterproof. Also, we could have desmosomes. Desmosomes are like spot welds or rivets that help hold cells together. So if there's mechanical stress, like a pulling action, the cells won't fall apart. Also, there can be gap junctions, little tiny tubes or cylinders that allow molecules to move from one cell to the next. So I like this slide because it kind of shows all of those things in one slide. Let's talk about cytoplasm. It's kind of a jelly-like substance that makes up a lot of the inside of a cell. It's not the stuff in the nucleus. It is the site where most cellular activities happen. And let's take a look at what's in there. So we've got cytosol, that's the fluid that suspends everything, and it contains things like nutrients and electrolytes. We have inclusions. These are the nutrients and electrolytes and cell products that are floating around in the cytosol. And then we have organelles. These are a little bit more complex. These are little machinery items they're describing here, which I agree. Little structures that actually do a function that are floating around in the cytosol. An example, for example, would be mitochondria. These little organelles, like mitochondria, have their own membranes around them, and they have their own little functions that they do. And we're going to take a look at those. What a great slide this is. Okay, let's take a look here. This is a cell. It is encased in a cell membrane, also known as a plasma membrane. It has a nucleus, which is wrapped in a nuclear envelope, which is its own membrane, and it has pores so things can move in and out of the nucleus. Also in the nucleus we have chromatin, which is the DNA, and we have the nucleolus. Okay, also we have some new structures here we haven't talked about. In green we have the Golgi apparatus. We have something called smooth endoplasmic reticulum. There is also going to be rough endoplasmic reticulum. It is shown here actually with red studs on it. We have mitochondria, they're kind of bean-shaped. These are the powerhouse of the cell where ATP are produced. We have lysosomes. Lysosomes are like bags of bleach, I guess I could simplify them as. They're little vesicles that contain digestive enzymes. We're seeing some centrioles in here, little rod-shaped structures. Microtubules and intermediate filaments, these give structures to the cell. Peroxisomes we're going to be talking about. And we have cytosol. Of course, cytosol is sort of the fluid that everything is floating in. Our cells need ATP for energy. So where do we produce our ATP? Well, it's in the mitochondria mainly. So we call them the powerhouse of the cell. The mitochondria have a very important role in aerobic metabolism, which creates ATP using oxygen and a few other things actually. A few other things are required, including glucose. So let's look at the mitochondria and the structure of it. It is double-walled, so it has two membranes, and it has cristae on the inner membrane. Also, it's bean-shaped. Ribosomes produce proteins. They can be floating freely in the cytoplasm, or they can be attached to endoplasmic reticulum, and then we call the endoplasmic reticulum rough endoplasmic reticulum. So it's kind of like studded into the endoplasmic reticulum. The endoplasmic reticulum is continuous from the nuclear envelope, and it's like a canal system that allows substances to be carried around. There are two types, rough and smooth. The rough is studded with ribosomes and is involved with the production of proteins. The smooth does not have ribosomes on it. It's involved with lipid metabolism and detoxification of drugs. The Golgi apparatus looks like another canal system, kind of flattened membranes. It has a lot of tiny vesicles around it. It sort of modifies and packages up proteins that are delivered to it by the rough endoplasmic reticulum, and it also produces packages of called vesicles and lysosomes. You can see the Golgi apparatus receiving a package from the rough endoplasmic reticulum. That package contains proteins. The Golgi apparatus is further going to modify and repackage these proteins. It can also do things like create little vesicles and little lysosomes. The cells use enzymes to help break down things. Some enzymes, like digestive enzymes, are stored in lysosomes. Other enzymes which are used to detoxify alcohol, for example, are stored in peroxisomes. So we've got lysosomes, which are kind of these bags of digestive enzymes. And also, the lysosomes can contain phagocytes. Phagocytes are like Pac-Man, if you've ever played that game. They're able to engulf bacteria and cellular debris. And then we've got the peroxisomes, which of course contain the enzymes which help to break down alcohol. So how does a cell maintain its structure or shape? It's by way of something called the cytoskeleton, not quite like the skeleton that we have for our whole body, but a little bit like that, I guess. Made up of protein structures that are listed here. They provide some shape to the cell. Centrioles are involved with cell division. The next 5 slides are a good review of what we've chatted about so far. There is quite a bit of additional information here, but I hope that it helps you with your review. This slide is heavily weighted on tests. If we look at the surface of some cells, they have strange little extensions. Cilia are like little hairs that move things across the surface, such as mucus in the respiratory system. Flagella help a cell actually move its entire self. The only example that we have is sperm, which is shown in the next slide. Microvilli are little up and down extensions of a cell that increase surface area. We know that cells come in all different types of size and shapes, and the following 10 or 12 slides or so show different versions of cells and the different shapes that they come in and how those shapes affect how they function. Next, we're going to talk about membrane transport. So how things are going to get across the cell membrane. This is very important. It actually guides how we choose IV fluids, for example, that we're administering to patients, how we give aerosolized 3% sodium chloride to some patients in order to induce expectoration of sputum. Anyway, I just want to relate that to clinical, how these next few things actually have a big basis of some of the therapies that we do. So we need to start off with a little bit of a basic understanding of solution. Solution is termed a homogeneous mixture of 2 or more components. That means that homogeneous means that it's sort of mixed equally throughout. So we're going to have solutes mixed equally throughout a solvent. So a solvent is the dissolving medium, and usually we think of water, for example, in our body. And then the solute are the things that are inside of that water or in that water to create a solution. Our easiest example would be sodium chloride dissolved in water. So we take a cup of water, we dump in some table salt, we stir it, and we see that it gets dissolved and we create a solution. The solutes are what we've added to the solvent, which is the water. If we look inside a cell, we see that there's nucleoplasm inside the nucleus, and there's cytosol in the rest of the cell. It contains things like oxygen, CO2, glucose, sodium, chloride. All of these things are in the water. Now let's look at outside of the cell. Extracellular fluid is also known as interstitial fluid. This is all the fluid that's outside of the cell, and there are all kinds of things out there, like glucose is out there as well. Hormones are floating by, neurotransmitters like acetylcholine, salts and waste products like urea and uric acid and carbon dioxide. Is such an important point on this slide. The cell plasma membrane is called selectively permeable. That means that some things can get through and others can't get through. Of course, we generally want good things to come into the cell, like nutrients, and we want bad things to be kept out of the cell, like uric acid. Talk about things moving across the cell membrane. If they don't require the addition of ATP to move across the cell membrane, we call them passive. If they do require the addition of ATP energy to move across the cell membrane, then they're called active processes.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, this type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. Cells use all these processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes.
It's an amazing video. I encourage you to watch it over and over again and take some really great notes as usual with these videos. It talks about the different types of transport. Now it gets into co-transport, which we don't tend to specify that type as much, so don't worry about that term. All right. Diffusion is a very important concept. It is how oxygen moves through our body and how CO2 moves through our body, so that's pretty important. So with diffusion, it's the idea of molecules moving from an area of high concentration to low concentration. It depends on a concentration gradient. The particles have a desire to spread themselves evenly throughout the air or solution, for example. Kinetic energy, which is termed the energy of motion, causes the molecules to move randomly in their environment. This is what leads to diffusion. Smaller molecules will diffuse faster, and higher temperatures will create faster diffusion. This is an awesome image of diffusion. We see that the molecules are becoming distributed evenly in the solution. Now, in terms of crossing a cell membrane using diffusion, we need to have the following. The molecules need to be small enough in order to pass through the membrane pores. The pores are created by those proteins. Remember how I described the proteins as olives that go all the way through a sandwich and they have a pore in the middle? Well, if the molecule is small enough, it might be able to squeeze through the pore. Also, if the molecules are lipid soluble, they're actually going to be able to diffuse more easily directly across the cell membrane. And if the molecules are assisted by a membrane carrier, some types of molecules will have a special membrane carrier that wants to carry them across the cell membrane. Simple diffusion, small lipid molecules are able to move through the phospholipid bilayer. This requires no addition of ATP, therefore it is passive. A certain type of diffusion called osmosis is the idea of water molecules moving from an area of higher concentration to less through some protein carriers on the cell membrane. They're called aquaporins. Here we see water molecules have a concentration gradient. In this case, there are more water molecules outside than inside the cell. So they are going to move by diffusion through a protein channel. This requires no addition of energy, so it is a passive process. We're going to talk a lot about osmosis because it affects the types of IV fluids that we will give to patients. So, the movement of water molecules depends on a concentration gradient, as we said in the previous slide. In the previous slide, we saw that there was a strong concentration or a higher concentration of water molecules outside of the cell. It is the idea that the water molecules were not diluted out by hardly any other substances, whereas in the inside of the cell, let's say that there were a lot of additional substances that kind of diluted out the water molecules. That made water want to move from the outside of the cell to the inside. So let's look at something called isotonic solutions. When we put a cell into a water bath, basically, of an isotonic solution, it means it has the same concentration of water, and no water will need to move in or out of the cell. What if we put the cell into a hypertonic solution? Hypertonic means that it contains more solutes and less water than the cells do. Now what will happen is that there's not much water outside of the cell, there's more water inside of the cell. So water is going to leave and go out through those protein channels, and the cell will begin to shrink or crenate. What about if we put a cell into a hypotonic solution? A hypotonic solution has less solutes and a greater concentration of water. So in the solution, there is a greater concentration of water than what the cells have. So water is going to want to go into the cell, and the cells will plump up or swell. The red blood cell in the left is in an isotonic solution. The water concentrations are equal outside of the cell membrane and inside of the cell membrane. Therefore, there's no movement of water, and the red blood cell maintains its shape. This is a perfect looking red blood cell, by the way, in image A. In image B, the red blood cell is submersed into a hypertonic solution. And what is supposed to be shown by this image here is crenation or shrinking of the cell, because there is a higher water concentration inside of the cell. So water leaves to go to outside of the cell because there was less water concentration outside of the cell. In image C, what we're seeing is a swelling of the red blood cell, and in fact it can actually burst when it is submersed into a hypotonic solution. A hypotonic solution has less solutes, higher water concentration than inside the cell. So the water is going to want to move into the cell and cause it to swell. This is all a type of osmosis. It is also considered a type of diffusion, and it is passive. It requires no additional ATP. When we've got some larger things that need to move across the cell membrane down their concentration gradient, we do that through protein channels. We do not need ATP added in this case, and we call it facilitated diffusion. See the protein channels in purple opening and closing, and they are allowing larger molecules or lipid insoluble molecules to move down their concentration gradient. I see it's shown in the image very well, and no ATP is required. Again, it's facilitated diffusion. Have you ever watched your grandmother make jelly and use one of those jelly bags? What she may have done is, boiled up some blackberries and put them in a jelly bag, and then squeeze that jelly bag by wringing it really tightly. This process would be called filtration. Our body uses the same idea in some parts of it. Water and solutes are forced through the membrane by fluid or hydrostatic pressure. So just simply increasing in pressure on one side causes water and small solutes to slip through the membrane. A pressure gradient must exist from an area of high pressure to lower pressure, of course, and filtration is used in the kidneys. So sometimes ATP or energy needs to be used in order to transport things across a cell membrane. This is called active processes, and active processes are used in all types of different situations, but they might include if a molecule is too large, or it's not lipid soluble, or we need to move substances against their concentration gradient, as in sort of like uphill. We need to push them into an area where they're already highly concentrated. Types of active processes we're going to look at are active transport across the cell membrane and vesicular transport. Let's start with active transport. It is used to carry some of the things listed here. We use ATP, of course, and we move against a concentration gradient in most of the cases. The perfect example is the classic sodium-potassium pump. It's going to be described in more detail on the next slide, but it is needed for nerve impulses, and it is brought up over and over again in this course. Oh, this is so much fun talking about this. So what we see in this image is the phospholipid bilayer with the heads and tails, and we see the proteins that, in these cases, they're spanning the entire width of the cell membrane. They're shown in purple. We're going to do an active transport here, and we're going to use ATP. So ATP needs to be added to the protein, and then the protein can do some work for us. It can pump out 3 sodiums against their concentration gradient and pump 2 potassiums inwards into the cell against the concentration gradient. This is a very important concept to get down pat. I encourage you to make some of your own simple notes for this process. It's called the sodium-potassium pump. And again, extracellular fluid, or the outside of the cell is at the top of the slide, and the inside of the cell is at the bottom. We're pumping out 3 sodiums, bringing in 2 potassiums, going against concentration gradient, and we're adding ATP. There are other ways of moving things in and out of a cell which use ATP. So these are active processes as well, and they're known as vesicular transport. There's exocytosis, which moves things out of a cell. Endocytosis moves things into the cell. 2 types of those: phagocytosis, which is solid materials being moved into a cell, and pinocytosis is like cell drinking, bringing liquids into a cell. This describes exocytosis very well, where materials are put into a little sac which goes out to the edge of the cell membrane and merges with the cell membrane. See that excellent example of exocytosis. In the bottom of the image, we see a vesicle moving out to the cell membrane and becoming continuous with the cell membrane and expelling its contents. An example of this would be seen at the end of a nerve, where acetylcholine can be released into the synaptic cleft to allow chemical messenger to go across the synaptic cleft and tell a muscle to contract. The term phagocytosis comes up over and over again in general anatomy and physiology. It is the idea that a cell is able to engulf particles such as another cell, like a bacteria, or parts of another cell. So that is a type of endocytosis. Another type of endocytosis is called pinocytosis, and that's engulfing some liquids and bringing them into the cell. The cell life cycle has 2 portions. Its regular phase, where it goes about its daily business, like growing and metabolic processes like producing things. But then it has cell division, where it reproduces itself. Keep this cell division pretty simple, so this is just a bit of an overview. You probably appreciate that it can be made very complex. In order for cells to divide, the DNA needs to get ready to divide. So the genetic material is duplicated, and now the cell is ready for division into 2 cells. So the way that happens is the DNA uncoils from itself and forms 2 nucleotide chains. Each side serves as a template, or sort of a base. And nucleotides are complementary. So the A always bonds to the T, and the G always bonds to the C. So an example here is that if we had the order of T, A, C, T, G, C, then it's going to bond with the complementary nucleotides. We can see that in the next image. Here we see DNA uncoiling and new nucleotides coming and joining onto the exposed nucleotides. They are joining on in the way that we mentioned in the previous slide, where A joins with T, G joins with C. Mitosis is cell division that results in 2 daughter cells, and there are the stages known as interphase, prophase, metaphase, anaphase, and telophase. They are described in the following slide. I urge you to watch the slide over and over again, the video, within the slide, and make excellent notes that describe these phases. Just also as a note, there's a different type of cell division called meiosis, which results in sperm or egg production. Moving on to protein synthesis. Sometimes our cells need some proteins to be built. So how does that happen? Well, the DNA serves as the map or the recipe for how to make proteins, and the DNA is stored inside the nucleus. A gene is a DNA segment that carries a blueprint on how to build one protein. So for example, in a recipe book, we would have recipes, we could think of them as genes, on how to make one recipe or one meal. So the gene has the blueprint for one protein or polypeptide chain. So the reason for all of this is that our cells need to build proteins because they function as things such as enzymes in reactions and building material. The interesting thing is that our ribosomes are very good at building proteins, and they are located out in the cytoplasm. However, the recipe or the DNA is not inside the nucleus and it's not actually allowed to leave the nucleus. So how do we get this recipe information or blueprint from the DNA, which is inside the nucleus, out to the ribosomes that are out in the cytoplasm? Well, we use something called RNA as a messenger. So the RNA, which is ribonucleic acid, is able to transcribe, or basically like write down, you can think of it as, the recipe and carry it out to the ribosomes. So then the ribosomes know how to build proteins. How do RNA differ from DNA? They are a little bit different. RNA is termed ribonucleic acid, and it is single strand, not double strand like DNA. It contains ribose sugars instead of deoxyribose, and the RNA contain uracil instead of thymine, the T that we referred to earlier. So they're a little bit different from each other. Very basic or simplified method of differentiating between three types of RNA. We've got transfer RNA, ribosomal RNA, and messenger RNA. So the transfer RNA actually transfer the appropriate amino acids to the ribosomes to help them build proteins. Ribosomal RNA help form the ribosomes in the first place, where proteins are going to be built. And messenger RNA are the type we talked about in the previous slide. They carry the instructions for how to build a protein from the DNA in the nucleus out to the ribosome.